that we know how we can create antibodies against nearly any type of antigen, today we're going to talk about certain research and clinical assays that involve those monoclonal antibodies. ELISA is an assay that takes place in a 96-well plate, which is what we see here. There are 96 separate wells, and the plate that's used for an ELISA plate, all of the wells are charged, so that the first substance we put into the ELISA plate wells is actually going to stick to the plastic. Now, what is an ELISA used for? It's used for research, but it's also used for lots of different tests in hospital laboratories. Most diagnostic tests to see if you were infected with a pathogen, they don't actually look for the presence of the pathogen. And that makes sense because maybe the pathogen isn't around anymore, but you still were infected, or potentially the pathogen isn't going to be found in the bloodstream, and you can't take out a piece of liver to test that if the pathogen's found in the liver. So blood is the easiest organ or tissue to isolate. And if we can't find the pathogen in the blood, well, we certainly can find antibodies against the pathogen in the blood. So, for example, if you get a blood test to see if you've been infected with HIV, that blood test doesn't actually look for the human immunodeficiency virus. It looks for antibodies against the human immunodeficiency virus. Because if you have antibodies against HIV in your system, then that means that your immune system cells have seen HIV and created antibodies against it. And if you've seen HIV, that means that you're infected with it. Okay, so most diagnostic tests to see if you're infected with a pathogen don't actually look for the pathogen, they look for antibodies as evidence of that infection. And a major way to do this is by using an ELISA. An indirect ELISA is going to look for those antibodies. We call it indirect because it's not showing that you're infected with a pathogen. You're not looking for the pathogen in this case. You're looking for evidence of infection, and that's why it's an indirect ELISA, because you're looking for antibodies and not the pathogen itself. So it uses an ELISA plate. It's a 96-well plate. And ELISAs never use cells, okay? So only supernatant, like the media around cells, or plasma, serum. It's only going to be liquid samples that are going to go into an ELISA plate. Now, I'm going to draw out the steps of doing an ELISA, and I think that it would be really helpful if you drew along with me so that together we can do an ELISA and you can see how the steps lead to the diagnosis of whether an individual has antibodies against a pathogen or not. So let's draw this out together. Let's draw out two wells of our ELISA plate, okay? And we'll put two different patient samples into each well. So in this case, we'll have the well on the left be the sample from an infected individual. Let's say we're testing for HIV. So the individual on the left is going to be infected, and the individual sample that goes into the right well is going to be uninfected. Okay, so let's go through the steps of how this ELISA works. Now remember, I told you that the first thing that goes into the ELISA plate is actually going to stick to the plastic of the ELISA plate because the ELISA plate has been treated, it's charged. So in the ELISA test, if we're going to look for patient antibodies, whatever those patient antibodies are going to bind to, that's what we're going to coat the wells with first. Okay, so in this case, we're going to look for antibodies that recognize HIV. Now, we're not going to put the entire HIV in the wells, but we're going to put HIV proteins in the wells to act as antigen. Okay, so step one is to coat the plate with the antigen. And the antigen that you're coating with is what the antibody is going to recognize. So if you're looking for hepatitis virus, you would put hepatitis virus proteins in the well. If you're looking for HIV, you put HIV proteins in the well. If you're looking for antibodies against a cytokine, then you would put the cytokine antigens in the well. Okay, so we know anything can act as an antigen. And if you want to figure out if there's antibodies against it, you just coat that antigen into the bottom of your ELISA plate. So that's the first thing. Let's draw our antigen into our plate. Okay, great. So we're going to let this antigen sit in there probably for about 1 to 2 hours, or even overnight in the refrigerator would be fine if you want to come back the next day. So our antigen is here, but there are still areas of that charged well that might still be sticky because the antigen isn't bound there. So we're going to rinse out the well, and then we are going to add in a solution into the well that has lots of proteins in it, just non-specific proteins. And all of those proteins are going to stick into the well. And essentially that's going to block the plate. We call it blocking the plate because it makes it so that whatever sample we put in next isn't going to non-specifically stick to the plate. It blocked any sites where proteins could stick to the plate. And we might use fetal bovine serum in a liquid to block the plate non-specifically. Another common blocking agent is just milk. We can actually put milk in the plate. Milk has lots of proteins, and they'll non-specifically bind up any areas of the plate that our antigen isn't bound to. Okay, so the next step is to block the plate. And so that's going to involve putting our liquid in there, and that's just essentially going to bind up any parts of the plate that are not already bound to our antigen that we're interested in. So the ability to create ELISAs like this is one of the amazing applications of an ELISA. We could look for antibodies against nearly any antigen. As long as we have some of that antigen, we can just coat it on the ELISA plate and it will stick to the plate. Okay, so we have ELISAs against hundreds of different proteins, different antibodies, and we use them a lot, both in clinical research laboratories, but we can also use this in research as well. So we've coated the plate with our antigen, which is stuck into the well. We rinsed out that solution, we added in our blocking solution. We allow that to sit for 1 or 2 hours, and that's going to block any areas that the antigen isn't stuck to. So we're then going to rinse that out. We rinse out that solution, and now finally we can add in the patient samples. In this case, the easiest patient sample to add in is going to be part of the blood. Now remember, I said ELISAs don't have cells in them, so after someone draws blood, in that tube, they're going to have to centrifuge that tube so that all the cells gets centrifuged to the bottom of the tube, and what's left is going to be the plasma or the serum. So we're going to take the serum of the individual, for instance, and we're going to add it into the well of the ELISA. And in that serum is where the antibodies are found. Antibodies are proteins, and they're too small to be centrifuged down with the cells. They're actually going to remain in the liquid serum of the blood. So we're going to add in our serum into the well, and here's the serum from that infected individual, and here's the serum from the uninfected individual that's going into the other well. Now within that serum, there's going to be antibodies, and in this case, remember, this ELISA recognizes whether there's going to be HIV antibodies, because it's an HIV antigen that's bound to the bottom of the well. So if the individual has antibodies in his or her blood that bind to HIV, they're going to bind to those HIV antigens that are bound in the well, and that's going to immobilize them there. So now, in this individual that is infected with HIV, he or she has antibodies against HIV, and lo and behold, they have bound to the antigens that are found in that ELISA well. So now we have those antibodies bound there. Now, of course, they're going to have other antibodies as well in the blood, but because those don't bind to HIV, they're not going to get stuck to the well. Now this uninfected individual, he's got antibodies as well, but you see none of them recognize HIV. They're antibodies against other things. We have lots of antibodies that are always circulating in our bloodstream, maybe against other pathogens, but this time no HIV antibodies in this individual. Okay, so after we add those patient samples and we allow them time to bind, which is at least usually 2 hours, then we're going to rinse out the wells. And what's going to happen is that those antibodies that don't bind, they're going to get rinsed out of the well. So all of the antibodies in the uninfected patient are going to get rinsed out. No antibodies will remain. But over here in the well with the infected person, the antibodies against HIV are going to stay stuck to the HIV antigens that are stuck into the well of the ELISA plate. Any other antibodies are going to get rinsed out. But here we have those antibodies that are now bound and they're immobilized there. So the next step is going to be to add in what's called a detection antibody. Okay, detection antibody. And that detection antibody is attached to an enzyme. And that's why we call this enzyme-linked immunosorbent assay. Okay, because we're going to have immune stuff, antibodies, absorbed or bound to the plate. And one of the last steps is going to use an antibody that is linked to an enzyme-linked antibody. So that antibody is actually going to recognize the Fc portion of human antibody. Okay, so our detection antibody is going to recognize the Fc portion of the antibody. And remember, it's conjugated to an enzyme. It's enzyme-linked. So if the patient had antibodies against HIV in this case, they're going to be bound to the HIV antigens, and they're going to be immobilized in the plate. In this next step, when we add in our detection antibodies, if there is an antibody there, our detection antibodies are also going to bind to that antibody, because our detection antibodies bind to the Fc portion of antibodies, of human antibodies. So over here in our infected sample, we have that detection antibody that has now bound to the patient antibody. Those enzyme-linked antibodies are also going to go into our well with the uninfected sample. But you notice there's no antibody there for those detection antibodies to bind to. The detection antibodies recognize the Fc portion of antibodies. And because there were no anti-HIV antibodies in that patient sample, there's nothing for the detection antibodies to bind to. So when we add in the detection antibodies, we're going to give it an hour or two, enough time for those antibodies to bind to any patient antibodies that are found in the sample. And then we're going to rinse out the well. And over here in the uninfected well, all of those antibodies are going to be rinsed away because there was no antibody in the well for the detection antibodies to bind to. In the infected well, however, there were HIV antibodies there, and the detection antibody has bound to those antibodies. So now the detection antibody stays behind in the well. So after these are rinsed out, the final step is to add a substrate. Well, what is a substrate? Substrate is simply the molecule on which an enzyme acts. Okay, so enzymes act on reactants. They help to accelerate reactions. And whatever that enzyme specifically binds to is called the substrate of that enzyme. And in this case, the substrate that we're going to add in, if it gets cleaved, it actually changes color. So if the enzyme is present, it's going to cleave the substrate and we're going to see color. So when we add in the substrate, we're going to add it into both wells. Now over here in the uninfected sample well, there's no enzyme because there's no detection antibody found there. So when we add in the substrate, nothing is going to happen to the substrate. But over here in the infected well, when we add in the substrate, well, the detection antibody is there, so the enzyme is there. And that enzyme is going to cleave the substrate. And when it cleaves the substrate, we're going to get color produced. So if we see that at the end of our ELISA, our well turns color, then we know that that's a positive result. And in this case, what does that mean? Well, that means that there were, in fact, patient antibodies that recognize the antigen the plate was coated against. Okay, so the infected individual, that well of the ELISA plate is going to change color, whereas there's not going to be any color found in the uninfected ELISA well. Okay, so let's go through this. Let me show you. I did a little animation so you can see the whole thing as a review. Here are our ELISA wells, and we start by coating the plate with the antigen. And again, remember, the antigen is going to be recognized by the antibodies that we're putting in here. So if we're looking for HIV, these are going to be antigens from HIV. If we're looking for hepatitis virus, these are going to be antigens against that hepatitis virus. Okay, so we can change the antigen depending upon what antibodies we're looking for, because, again, we're doing an indirect ELISA, and an indirect ELISA is going to look for antibodies. So we add the antigen, and that's going to bind to the ELISA plate, because remember, the plate has been treated. We rinse out any antigen that's not bound, and then we block the plate. And that's going to fill in any areas of the wells so that our next sample that we add in isn't going to non-specifically stick into the well. After we rinse out that blocking agent, which is usually just milk or fetal bovine serum and some PBS, then we add the patient's samples. And in this case, here's that infected patient, and unfortunately, he or she has antibodies against our antigen. In this case, it's part of an HIV protein. Here's the uninfected patient. This person has never seen HIV, and so his or her immune system hasn't created antibodies against HIV. And we allow time for the patient's samples to bind, and then we rinse out the wells. Now you'll notice that the patient antibodies, they stayed stuck to the HIV antigens in that well. But all of the other antibodies from the uninfected patient were rinsed away because he or she didn't have any antibodies that recognize HIV. Now remember, the next step is going to be to add in that detection antibody. And the detection antibody is linked to an enzyme, enzyme-linked. So the detection antibody recognizes the Fc portion of human antibodies. So if there is a human antibody in the well, it's going to bind to it. You can see there's nothing in the uninfected patient well for those enzyme-linked antibodies to bind to. But in the infected patient well, because there are antibodies bound there that recognize the HIV antigen, the enzyme-linked detection antibody binds to that human patient antibody. Now we rinse out the well, and we can see that that detection antibody has remained behind in the well of that infected patient. Now the last step is to add in the substrate. And if the enzyme is present, it's going to cleave the substrate, and the substrate is going to turn color. So the substrate is just the particular molecule that that enzyme acts upon, and it's dissolved in a liquid solution, so we can add it into the well. So we add the substrate solution into the well, and if the enzyme is there, then it's going to cleave the substrate, and we're going to get color. Okay? Now, if we get color, that means that that is a positive sample, that there were patient antibodies there that recognize the antigen we used to coat the plate. In an uninfected patient, there's not going to be any color produced. And so here's an actual ELISA plate that shows the color. And you can see that there are various degrees of color that's formed. There's some lighter green, there's some darker green, there are some that are very light, and there are some that are completely clear. Now that is a qualitative assay at the top where we can just see whether it's yes or no. Is there color or is there not color? But we can use a spectrophotometer, which is the machine at the bottom, to figure out exactly the degree of color, okay, the intensity of the color that's found in that well. So a spectrophotometer is just going to look at the degree of intensity of a particular wavelength of light. Remember that colored light is just different wavelengths of light. So the spectrophotometer is going to be able to measure the intensity of these different colors. So we can put the ELISA plate into a 96-well spectrophotometer, and it will read how much color is found in every single well. And so that's the way that we can take a qualitative assay up at the top and turn it into a quantitative assay where we actually get a number for how much intensity is found there. Okay, so in that way, we can actually compare the samples to see if somebody has a lot of antibody against HIV and would have a very dark well, or maybe just a little bit of antibody against HIV, and that would be a lighter green well. So this is an indirect ELISA. An indirect ELISA measures antibodies. We also, though, can measure antigen using ELISA just by modifying the assay a little bit. And when we're measuring the antigen itself, we call that a sandwich ELISA or a direct ELISA because we're looking directly for the antigen. We're not looking for antibodies against the antigen, we're looking for the antigen itself, so it's a direct ELISA. And we call it a sandwich ELISA because we actually make a little antigen sandwich where the meat is the antigen and the bread is made of antibodies. So if we're going to detect an antigen, instead of binding an antigen into the ELISA well, we bind antibodies. So we call this our capture antibodies. we bind our capture antibodies, and this is going to specify what the ELISA is going to measure. Because next we're going to add in our patient samples. And if there's that particular antigen that we're looking for, it's going to bind to these antibodies. So let's say that we're looking for some cytokine found in a patient's blood. Maybe they have rheumatoid arthritis and they're producing too much TNF. So we're going to do an ELISA that looks for TNF. And we're going to start by binding our capture antibodies to the plate. These capture antibodies are going to bind to TNF, so they're anti-TNF antibodies. So we bind those first. We're going to do the same process as we did before. We're going to allow time for those to bind. We're going to rinse them out. We're going to block the plate so there's no non-specific binding of the patient sample. We're going to rinse that out, and then we're going to add the patient sample. So here we've added the patient sample, and as you can see, this patient does have some TNF found in her blood. The TNF has bound to the anti-TNF antibodies that are on the plate already. So we allow time for the binding to occur, and then we rinse out the patient's sample, and then just like we did before, we add a secondary antibody, which is a detection antibody. And in this case, the detection antibody is enzyme-linked, just as it was before, but instead of recognizing the Fc portion of an antibody, it recognizes the antigen that we're measuring here. So we coated the plate with an antibody that recognizes TNF. The TNF bound to those antibodies, and so now the detection antibody is also going to recognize TNF. Okay? The only difference between the capture antibody and the detection antibody is that the detection antibody is linked to that enzyme. Okay, so we add in our detection antibodies, which are enzyme-linked. We allow time for binding to occur, and then we rinse out the well, and then finally we add our substrate. And if those detection antibodies are still present because they bound to the antigen that was bound to the capture antibodies, then that enzyme is going to cleave the substrate and we're going to get color, just like we did in the indirect ELISA. Okay, now you can see why this is called a sandwich ELISA, because we end up making a little sandwich where the bread is actually antibodies. So to summarize, an indirect ELISA is going to measure antibodies. A sandwich ELISA is also known as a direct ELISA because it measures the antigen. And the difference between these is really just what is coded on the ELISA plate at the very beginning. If you code the antigen, well, that means you're going to be assaying for the next thing that comes in, which is going to be the patient sample. And it's going to be antibodies found in the patient sample. But if you coat the ELISA plate with an antibody, well, then that antibody is going to bind to some antigen that's found in the patient sample. Okay, and then the detection antibody and the substrate steps are exactly the same. Now that is an ELISA.